It is actually really a great pleasure too for me to host today's talk. Now, before I introduce our two speakers, I wanted to say a few words. So we all know, as most of the people here working on life science, protein synthesis, therefore tRNA synthesis, every process involved in living organism will need tRNA synthesis. So here we have pioneers who started the field. Professor Paul Schumer, many of you have heard him speaking on the tRNA synthesis, and two of his proteges, and with us. You know, over many years, they have been decoding how these family of important enzymes work in different forms of life. And recently, they discovered that a totally new universe of the functions of this ancient family of enzymes. And perhaps not surprisingly, because the family of enzyme is related or involved or critical to any forms of life process. Therefore, the family of enzymes is not only involved in normal physiology of human beings, but also on many of the diseases involved. So we will be hearing some of them. But that brings me really tremendous amount of pleasure to introduce our two speakers. Professor Susan Martinis from University of Illinois in Urbana-Champaign. Professor Martinis graduated from University of Illinois, and did a postdoc with Paul Schumer when his lab was still in MIT. I won't say which year, but very young, very recent, I think. Okay, and after postdoc, so Susan was hired as the third person in HKUST Pharmaceuticals. Some of you might know that the company was bought by Merck at a price of $9.1 billion. Okay, so I was saying that Susan, if she still holds some of the original stocks, you can imagine, right? Susan moved back to academic in University of Houston and rose to full professor, but I guess she still loves alma mater. So went back to UIUC some years ago. So she has been chair of the Department of Biochemistry for over the 5-6 years, right? And the department is a wonderful department. I looked into the portfolios of wonderful biochemistry, biophysics, structure and functions of many critical biological. So I will not say too much, otherwise I will be using too much of the time. And the dynamic duo will be presenting very continuously without a break. So I thought I introduce our second speaker now rather than in the middle. So that allow me to introduce Professor Karin Musier-Forsyth, who graduated from Cornell University in Canada, if I remember correctly. And then also joined Professor Paul Schumer's lab postdoc, together with, I think, very closely together, it's almost same time with Susan. And she very successful postdoc and established her own lab in University of Minnesota. In University of Minnesota for more than 10 years, many prestigious titles, wisdom, professor, others. I guess Minnesota is too cold, right? So Karin decided to move to Ohio, where she is the Ohio eminent scholar. And they will be telling us their wonderful works on tRNA synthetase, which are related to different forms of life. I will not repeat the titles in here.
Thank you, Ming-Chi, for your hospitality. Very warm welcome. I must confess that Karin and I have worked to be really efficient today, because what we'd really like to do is, Ming-Chi is giving a seminar at 4 o'clock, and so we were trying to be efficient so that we can also make that. I want to thank you and the audience for being here. I just want to, this is our third trip to Hong Kong, and we've just enjoyed the warmth of the people here, the students at the university, the faculty and their hospitality, the fine meals. It's just been a wonderful experience, and we look forward to coming back again and again and again. Thanks for talking about my original experience at HKUST Pharmaceuticals, and I was lucky to be in Paul Schimmel's lab at that time when he set that company up. And it was founded originally to identify new antibiotics that would target tRNA synthetases. And I eventually, within a few years, I went into academia from that experience. With my husband and I moved down to Houston to take on assistant professor positions. But what I took from HKUST is this idea of doing basic biomedical sciences and thinking about long-term applications. And I think that's what our title envelopes or comprises today, is Karin and I want to talk to you about, are some examples of basic biomedical research that really have gone on and have been useful in the clinics to help people with their health and in many different health issues. So our story begins at this wonderful family of enzymes, the tRNA synthetases, which are responsible for setting the genetic code in the first step of protein synthesis. So it's here where tRNA synthetases take a specific amino acid, attach it to a tRNA, and once this amino acid is linked to an adapter tRNA molecule, it's shuttled to the ribosome where it's incorporated into the growing polypeptide chain. All tRNA synthetases have a two-step reaction in which they utilize ATP. They activate the amino acid so that it can be transferred to the tRNA in the second step. And what I want to emphasize about this particular reaction is it occurs in the ancient core of the tRNA synthetase. Now, the tRNA synthetases have evolved to be very large. They have multiple domains, some of which I'm not showing in this crystal structure. But just a small part of what this enzyme does, the synthetic reaction in which it charges tRNA with an amino acid, critical to protein synthesis, occurs just in a very small region in this ancient core of the enzyme. Okay, but nature and humans have taken advantage of these enzymes to develop therapeutics that act as antibiotics. So Bactroban is a cream that was marketed originally by SmithKline Beecham. It's been around for decades. And it's used if you had a skin infection, a Staph aureus infection, you might have used Bactroban or mupirocin to treat that infection. Its active ingredient is pseudomonic acid. It was isolated in the 1960s, a natural product from a bacteria called Pseudomonas fluorescens. And when it was discovered that it was a potent antibiotic against gram-positive bacteria, it was developed into this antibiotic that's marketed as Bactroban, mupirocin. Later, they discovered that it was an inhibitor of a tRNA synthetase, isoleucyl-tRNA synthetase. And Tom Steitz solved the crystal structure of isoleucyl-tRNA synthetase bound to this antibiotic. And you can see isoleucyl-tRNA synthetase is in many colors here. It's a very large molecule. The different colors indicate different domains. The tRNA, right here, binds along a large interface of it. But if you look very closely here, the pseudomonic acid just binds a little part of it, and that's that ancient core. It binds that ancient core in a very species-specific way, capitalized upon in the diversity of the tRNA synthetases and the divergence between bacterial and human isoleucyl-tRNA synthetases to be an effective antibiotic. A second example that I'd like to share is an ancient herb that's been used as a Chinese medicine. And this ancient herb was found in hydrangeas, or the root of hydrangeas, the chansang root. Its active ingredient is febrifugin, and febrifugins is used to treat the fevers of malaria and to bring those fevers down. So it's been known for 2,000 years that this root carries this remarkable activity to help malaria patients. It was recently that Paul Schimmel and Xianglei Yang's groups both have faculty positions at the University of Hong Kong Science and Technology, solved the crystal structure of a synthetase, prolyl-tRNA synthetase, with ribofugin bound to it, and found that it also bound to that ancient synthetic site on the prolyl-tRNA synthetase, interrupting its interactions with the substrates ATP and proline. Now, there's up to 20 different tRNA synthetases in every cell, in every organism, in all kingdoms of life. These tRNA synthetases are specific for each one of the amino acids that are represented in proteins. So prolyl-tRNA synthetase would activate proline. Isoleucyl-tRNA synthetase would activate isoleucine. And what this provides with these two proofs of principle that I've shared with you, a remarkable array of targets that pharmaceutical companies have capitalized upon for years to try and develop new antibiotics, antifungals, and other agents. Now there's a very, I've been talking about these synthetic active site interactions, and if you interfere with that and you can't activate that amino acid, you cannot charge that tRNA, then the cell will die. I want to talk about another area of the tRNA synthetase that's more recently been capitalized upon as a new target site within the tRNA synthetase to develop a very potent antifungal that's being used for new leads all over this pharmaceutical industry to develop new medicines. So the tRNA synthetases are challenged with very closely related looking substrates, so they can make mistakes. But they've evolved mechanisms and extra domains to help curb their mistakes that allow them to have a fidelity that with an error rate of up to 1 in 10,000, which is quite high. And it's thought that this high fidelity is critical to the cell survival and cell maintenance of the basic processes that are required for a healthy cell. And so these high fidelity, carefully charged tRNAs then are transported to the ribosome where they're incorporated into the polypeptide chain, as I said before. So how do the tRNA synthetases maintain this high level of fidelity? Well, about half of them have an editing mechanism. And this editing mechanism was first proposed by Sir Alan Furst at Cambridge University in England. And there, in the 1970s, he had a sort of a brilliant conceptual idea of how these might work. And all the work that we've done since he first proposed this has shown that this is true. So basically, the synthetases that require it, and it's about half of them, have two filters. The first filter is kind of a coarse filter. And it binds to a set of amino acids. Some of them are the wrong amino acids, but they're closely shaped or structurally similar to the cognate amino acid. So they're activated. Well, then these tRNA synthetases transfer those activated amino acids to a second filter. And this filter is what we think of as a fine sieve. It has a different strategy for interacting with these activated substrates. And what's most important is the second site repels the correctly activated amino acid because that's the one you want. And it repels it, and that amino acid is charged to the tRNA, and then it goes off to the ribosome where you want it. It does bind to the misactivated amino acid and hydrolyzes it in this two-step reaction. So as we've gathered more tRNA synthetase crystal structures, we've found these two domains. This domain here where the tRNA is bound is that synthetic site in the ancient core of the enzyme. And this domain up here for leucyl-tRNA synthetase or isoleucyl or valyl-tRNA synthetase is called the CP1 domain. It was first coined by Paul Schimmel. The full name is connective polypeptide 1. And what we found years later is that it has a hydrolytic editing site and enzymatic activity that cleaves incorrectly charged tRNAs so that you can preserve the fidelity of the genetic code. So I'm going to be mainly talking to you about leucyl-tRNA synthetase. Leucyl-tRNA synthetase is challenged by several amino acids. That includes amino acids that are incorporated into proteins, such as methionine and valine. It also includes non-standard amino acids that aren't found in proteins, but are metabolite intermediates, such as norvaline or methionine. These can be activated, and then if they are activated, then they go through this charging reaction on the top. And if they're misactivated, then they undergo this post-transfer editing activity to release the amino acid and then avoid mistakes. We have a lot of crystal structure information that's available for leucyl-tRNA synthetases and other tRNA synthetases. So this particular crystal structure is the amino acylation complex. What I want you to notice here is that this end of the tRNA is diving into this ancient synthetic core. And it was that core where we saw, remember, that the Bactroban molecule, pseudomonic acid, was inhibiting. The editing complex, which was solved by my collaborator, Stephen Cusack, is here, and the tRNA is interacting with that. So it's a different active site and potentially another target that we can rely upon that carries out post-transfer editing. So now I want to talk to you about a set of inhibitors that was learned sort of fortuitously to interact with this editing site. And as of last summer, they've been developed into a potent new antifungal. So Anacor Pharmaceuticals was started by Stephen Benkovic at Penn State University and Lucy Shapiro at Stanford. And their idea was to make a series of small molecules that contain boron and screen them for bioactivities or inhibitory activities against panels of microorganisms. So they did that, and they found in fungal systems a particular boron-containing compound called AN2690 that inhibited leucyl-tRNA synthetase. And here I show an amino acylation assay in which we're just looking at the charging of tRNA. If you add this small molecule, then the amino acylation activity is inhibited. As they developed this and found they're looking at a potential new antifungal, called it tavaborole. Most recently, it was marketed as Kiridin to treat onychomycosis, which is toenail fungus. That was last summer. So what was interesting is that this is inhibiting amino acylation activity, but we found out that this molecule was interacting with the editing site. Now you might imagine that you have a synthetic reaction that's essential for all living organisms. You have to have leucylated tRNA for cells to maintain and grow. But with editing, you could inhibit that to some extent and still have some cell survival. So what they found when in a collaboration with Stephen Cusack is that when this boron compound bound to the enzyme, it bound into the CP1 domain, and it basically lied in wait. It was waiting for the tRNA to come along, and when that tRNA came along, it cross-linked it in a unique reaction that involved the cisdiol groups, 2 hydroxyl groups on the ribose at the end of the tRNA, and this boron compound. It cross-links it where the boron forms 4 bonds now and interacts with the tRNA in a covalent way. And when it did this, it trapped the tRNA on the enzyme. It couldn't turn over, it couldn't leave, and it kind of took the tRNA out of commission. So here, this AN2690 binds in the CP1 editing domain, and then it locks down this editing conformation and the tRNA stuck to the enzyme, can't turn over, and it becomes a very potent inhibitor or antifungal in treating diseases. My lab has long been interested in the editing reaction, so we worked with Anacor on trying to understand this drug and the mechanistic implications and also resistance mechanisms. And I want to share with you one story that was carried out by my student, Hanchao Zhao, when they isolated a lysine residue that was resistant. And this lysine residue is very conserved across the eukaryotes, but it surprised us. We had done lots of mutagenesis in the editing site and felt that we understood all the key players that were involved in this post-transfer editing reaction. But what was unusual about this lysine was it was located quite far away, distal to the editing site, which is over here. So we were curious about this, and we carried out some enzymatic assays to examine the particular resistance mutant, and we also included some additional mutations, and we found that it carried out amino acylation activity as we would expect. When we added the inhibitor in vitro test tube reactions, that would, we found results that would correlate to the resistance of the organisms to the AN2690 in the presence of this mutation. And that is when we added the AN2690 with the mutant, we saw a rescue of the amino acylation activity. So the cells could continue to amino acylate tRNA and survive. We also looked at an editing reaction that occurs in that CP1 domain, and we found that this represents post-transfer editing where the tRNA is being hydrolyzed away from the amino acid. And when we introduced these resistance mutations, we saw some effect, but not a profound effect on editing. So we were curious about the mechanism and what this distal mutation was doing in the editing site to support editing and also proffer resistance to this very powerful new antifungal. My student Hanchao Zhao went to the literature and he looked at a crystal structure that was solved by Stephen Cusack of Candida albicans that was bound to one of these boron-containing compounds. And when he looked at the active site, he noticed two tyrosine, a tyrosine residue that Stephen Cusack had flagged in that publication that were in two different rotamer forms. Now, these tyrosines are near the lysine that we've been talking about. So we wondered if those would have some impact, those two rotamer forms, and the dynamic ability of that tyrosine residue would have some impact on editing and also the resistance. So Hanchao took the resistance mutants and also the AN2690, and he crystallized the complex. So here we see the crystallized form of the resistance mutant. This is where the lysine would be, and this is the tyrosine. And in this case, rather than seeing a mixture of the single amino acid in two states, he found that it was locked over in one state when that lysine residue was missing. So it was pushed over or pointed toward the lysine residue. And so what we hypothesized then is this rotamer, or these mixed states of the tyrosines, would play an important role in editing and also interactions with the antibiotics. So we collaborated with in Urbana with Zan Schulten, and Hanchao used some sophisticated computer modeling to build use his structures to build a complex that included the substrate intermediates for editing, and also that included the drug AN2690. And he subjected those to molecular dynamics and also metadynamics. And what he found when he followed that tyrosine is that it actually had three states. And those three states showed that the tyrosine would interact in different ways with the substrate itself. In one case, it would interact with the ribose hydroxyl, which we think is the active form post-transfer editing. In another case, it interacted with the phosphate group, which is a short-lived intermediate state. And in the third case, it would be pointed toward this lysine residue and also a phenylalanine residue, and we called that the inactive state. So it's essential that that tyrosine is pointed over toward the substrate to carry out editing reactivity, and that also I want to make a point that these states are reversible. So when we introduce this resistance mutation in which the lysine is changed to an alanine, it creates this deep energy well, and the tyrosine gets stuck, pointed over to that lysine residue. So Hanchow developed a mechanism for editing in which he showed that the tyrosine residue, when the substrate was bound, it switched rotamer states, and then the substrate settled in and could interact with the ribose hydroxyl of the tRNA and capture it for editing. And this would be the same sort of mechanism that helps capture the drug in this leucyl-tRNA synthetase editing site. And then the bond is cleaved, and the enzyme recycles itself. And then the tyrosine is restored in its resting state to its previous position and where it's interacting with the lysine residue. So what's important about this is that we understand how the antibiotic works and also the ways that the organism and the leucyl-tRNA synthetase in that organism might mutate to become resistant. But also what this work showed, and I don't have time to go into all the details, is that there was a big difference in the eukaryotic leucyl-tRNA synthetase editing mechanism and also the bacterial leucyl-tRNA synthetase mechanism. So the eukaryotic system, which I've been talking about, relies on this tyrosine residue. And it comes in and is perfectly positioned for this editing reaction. But if we go to the bacterial leucyl-tRNA synthetase, it's substituted by a threonine residue. And the threonine residue is actually on a completely different alpha helix in the protein. So there's a big conformational change. And why that's important is it offers diversity for us to develop new antibiotics. And there have been many that have been coming out of Anacor Pharmaceuticals that will interact with bacterial leucyl-tRNA synthetase in a specific way that doesn't inhibit human leucyl-tRNA synthetases. So at this point, the pharmaceutical industry has taken these boron-containing compounds that target and inhibit leucyl-tRNA synthetases and have been capitalizing upon them to go far beyond the antifungal activities and develop whole new generations of antibiotics. I think I've talked about the people that have contributed to this work. And so at this point, I'd like to kind of segue in our discussion or presentation about the different therapeutic activities by targeting tRNA synthetases and move along to what Karin is going to present next, and that is potential applications as an antiviral. So thank you.
I also want to echo what Susan said at the beginning and thank Minji and all the faculty and people that we've interacted with here, and of course Paul for giving us the opportunity in Hong Kong. It's great to be back, and I'm going to do part 2 here, which is focused on unexpected connections of synthetases to HIV. So as you all probably know, HIV is still a really important global health problem. So what this map of the world shows is the percentage of people, this age group, living with HIV in 2013. And so you can see a large concentration. It's of course still in South Africa, but it's still a worldwide global health problem. So 35 million people are living with HIV, 2 million people were newly infected with HIV in 2013, 1.5 million people died of AIDS, and 39 million people total have died of AIDS since the beginning of the pandemic. So a very important advance in treatment of HIV came in see, came about in 1996. And this was the advent of HAART, which stands for Highly Active Antiretroviral Therapy. And you might also know this as sort of the combination drug cocktails, triple cocktails. So when these started to be used, so the drugs in these cocktails target different targets. I'll talk about the targets in a minute. There was a rapid, in the United States where the drugs were available, a very rapid decline in the number of deaths. And this graph here shows what happened globally. And so, as you can see, there's now a decrease in the number of newly infected and also the number of deaths, but there's a steady increase in the number of people living with HIV. And that's because HIV is now really just a chronic illness that has to be handled by antiretroviral, that can be handled by these antiretroviral treatments. And a big problem with these, of course, is that there's not a single antiviral drug that has not led to some drug resistance problems. And so these patients have to continuously be changed in their therapeutic regimen. So because of this, there's still a need for new targets and new antiretrovirals. And what I want to explain with this slide is go through the life cycle briefly and show you where the current drugs are targeting. And so this is representing a virus which enters the T cells via certain receptors. Once inside the cell, there is an uncoating event which happens along with reverse transcription. So the hallmark of a retrovirus like HIV is that its genomic material is RNA, and that RNA has to be reverse transcribed into DNA before it can be integrated into the human genome. And after integration, at some point, the virus uses the host cell machinery to transcribe new viral RNA. There are two pools of RNA. One pool is used to translate viral proteins, and the other pool is used to be packaged into new viral particles. And exactly 2 copies of the RNA genome are packaged into the budding virion. The initial product that's made is an immature viral particle that's not infectious, and then there's a maturation process that results in a total rearrangement of the viral proteins and the infectious virus particle. So the current HIV drugs target 4 major steps in the viral life cycle: entry or fusion of the cell with the receptors, and then the 3 primary enzymatic steps: reverse transcription, so reverse transcriptase; integrase, which is responsible for integrating the viral, proviral DNA into the human genome; and then protease, which is responsible for viral maturation. And so there are currently no drugs, for example, that target, you see there's a lot of gaps in this cycle, and specifically there are no drugs that successfully target the assembly process, nor are there drugs that target processes that involve specific host cell proteins. And so the retroviruses use many tricks, and we could have a long list here of clever things that they do. One I've already mentioned is that they can easily lead to drug resistance, and that's because they mutate very rapidly and recombine very effectively. So they're constantly resulting in novel genotypes which can escape the action of drugs. Another trick that I just want to, again, emphasize is that they take advantage of a vast variety of host cell components. So it's been estimated that over 250 cellular proteins either facilitate or restrict retroviral infection. And the one that the host cell factor that I really want to focus on today is a cellular tRNA molecule, which the retroviruses use for a very unique and different function than they do in the than normally used, and I'll go into that in a second. And so the question is, can these host cell interactions be exploited as novel therapeutic targets? And in particular, what I'm going to focus on today is the potential for exploiting the role of the tRNAs. And so as Susan told you, the main function of tRNAs and an essential function for all living organisms is the amino isolation function. And so the tRNAs are specifically amino isolated or charged with specific tRNA molecules, which then go on and interact with the ribosome and deliver the amino acid to the site of protein synthesis. And what HIV and other retroviruses have done is recruit these tRNAs out of this pathway into new viral particles and use them for a completely different function. That function is outlined here in the next slide. So in this slide, the black represents the viral RNA genome, which again has to be reverse transcribed or converted into double-stranded DNA. And as with all DNA polymerases, reverse transcriptase needs a little primer to get that process going. And the primer that it's recruited for this function is the host tRNA. And so in the case of HIV, the 18 nucleotides at the end of human lysine tRNA3 are perfectly complementary to a region of the genome known as the primer binding site. And reverse transcriptase then recognizes this initiation complex and synthesizes the cDNA using this overhang here as the initial template. Reverse transcriptase has an RNase activity where it will degrade the RNA part of the new RNA DNA hybrid that's formed, leaving this overhang. There's then a strand transfer event, and then the rest of the so-called minus strand DNA is synthesized. There are multiple steps here I'm not showing, but eventually you get the full-length proviral DNA, which is then competent for integration into the human genome. So even though HIV can rapidly mutate and generate new sequences, new genotypes, the one region of the genome that is highly conserved and actually strictly conserved in all HIV patients that have been sequenced to date is the primer binding site. And so this region is never mutated. And that really shows you how critical priming is for the life cycle of HIV. So we thought that maybe understanding this process, how the tRNA is recruited away from its normal pathway in the cell, could potentially be exploited for a new therapeutic target. And so that's the first question I want to address today, is what diverts cellular tRNA from its normal function to be used in HIV. So it was shown number of years ago now by Larry Kleiman's group that whereas 6% of cytoplasmic tRNA is lysine specific, 60% of the tRNA found in HIV particles is lysine specific. So there are 3 different human tRNAs in the cell. These are shown here. They're called lysine 1 and 2, which are usually lumped together because they only have a single base pair difference in the anticodon stem here. And then tRNA lysine 3. This is the tRNA specific for lysine that has the perfect complementarity to the primer binding site, and this is the one that's used as a primer. However, what Larry Kleiman and his group showed is that all 3 of these human lysine tRNAs are abundantly packaged, selectively packaged, and represent the same distribution that you find in the cytoplasm. So this led us to, and this happens despite the fact that HIV really only needs tRNA lysine 3, but all of these tRNAs are packaged. And so this led us to hypothesize that maybe a cellular factor that's specific for all 3 isoacceptors is actually involved in this selective packaging process. And of course, the cellular factor that meets that is lysine tRNA synthetase. So this is the only factor that specifically has evolved to recognize these 3 tRNAs. And so and as Susan told you, again, the function of aminoacyl-tRNA synthetases, the normal function is to attach an amino acid to a specific set of tRNAs and then to deliver those to and then those are delivered to the site of protein synthesis at the ribosome. So we collaborated with Larry Kleiman to see whether, or to test actually, whether human lysine tRNA synthetase, along with its tRNA isoacceptors, is actually packaged into HIV particles. And what we found is that of the 9 synthetases we tested, human lysine tRNA synthetase was the only one that we found to be packaged into the particle. That this packaging requires a viral protein known as GAG, and we went on to show that there's a specific interaction between human lysine tRNA synthetase, shown here, bound to the tRNA, and the capsid domain of GAG, shown in red. Furthermore, what's important is that this is important for the viral life cycle. So if you knock down the expression of human lysine tRNA synthetase, that reduces HIV infectivity, and if you overexpress it, that actually enhances viral infectivity. So we knew this was important for the viral life cycle. So this led us to propose that the tRNA is recruited into the virus particle in complex with lysine tRNA synthetase through a specific interaction with the HIV gag protein. And we know that the tRNA is recruited at the assembly step rather than later on after the virus infects the cell. And so, so now we wanted to, of course, lysine tRNA synthetase has a very important canonical function in the cell. And to understand how it recruits the tRNA, we have to understand how it is actually diverted from that canonical pathway. The other thing that we have to realize is that in higher eukaryotic systems, 9 of the aminoacyl-tRNA synthetases, including lysine tRNA synthetase, are assembled into a multi-synthetase complex along with 3 other cellular factors. And so lysine tRNA synthetase is one of these synthetases that's part of the multi-synthetase complex. And it's believed that in higher eukaryotic cells, this multi-synthetase complex is, in fact, a way of compartmentalizing tRNA synthetases for their canonical function in translation. But in recent years, it's been shown that almost all of these aminoacyl-tRNA synthetases that are part of this multi-enzyme complex function in other pathways, in non-canonical pathways that are not related to translation. And so I'm not going to have any time to go through most of these arrows here, but just wanted to highlight the wide variety of pathways and functions in the cell that these synthetases are involved in. And as Minji said, dysregulation of these processes can result in human disease. And so I just want to point out on the next slide the pathways specifically that lysine tRNA synthetase has been shown to be involved in. So Sung-Hoon Kim's lab has shown that when the cell is stimulated by specific stimuli, for example, laminin, lysine tRNA synthetase can be phosphorylated, and that results in release of the synthetase from the complex, migration to the membrane where it interacts with the laminin receptor, and this has important implications in cancer. Ehud Razin's lab has shown that IgE stimulation of mast cells results in phosphorylation of lysine on a different site, on serine 207, which results in nuclear import and synthesis of AP4A and transcriptional activation of certain genes. Sung-Hoon Kim's lab has also shown that another phosphorylation event can happen that results in secretion of the synthetase and involvement in this signaling pathway. And what we have been investigating is the pathway by which lysine tRNA synthetase might be released from the complex and participate in the HIV assembly and reverse transcription. And so in order to understand this mechanism by which this might occur, we first wanted to ask, does HIV infection actually result in different levels of lysine tRNA synthesis expression? So there's actually a database that exists now that has monitored expression levels via messenger RNA levels of genes and how they change upon HIV infection. And what we see when we look at lysine tRNA synthetase is basically there's very little change from the normal level of expression in uninfected cells versus in infected cells. So HIV doesn't seem to be modulating this process by changing the expression levels of lysine tRNA synthetase. Of course, this is at the mRNA level. We also wanted to see what happened at the protein level, and we showed in 2 different cell types that basically the same result, that we get very little change in the expression of lysine tRNA synthetase during the course of HIV infection. So the next question is, since most of the non-canonical functions that have been well characterized have involved post-translational modifications of some sort, we wanted to know whether lysine tRNA synthetase might be phosphorylated following HIV infection. So this slide shows a Western blot where we're probing with 3 different antibodies, and we have done a pull down of lysine immunoprecipitation of lysine tRNA synthetase from uninfected cell lysate and then from infected cell lysate, HIV-infected cell lysate. And what we see is that, indeed, only in the case of HIV infection do we see a reaction with the phosphoserine antibody. And this is the pull down of the Western blot of Leu RS. And then this is GAG, which again we only see in the presence of the infected cell as expected. This is pulled down together with lysine-tRNA synthetase interact. So I have this blot was a little bit unclear, so I just have another independent experiment which was done to verify the phosphorylation of lysine-tRNA synthetase following HIV infection. So in many cases, these post-translational modifications will then alter the localization state of the synthetase. And so that's the next question. Does cellular localization change upon HIV infection? So what I'm going to show you on this next slide is confocal immunofluorescence images using confocal microscopy, where we're looking at human T cells, and the blue is the DAPI stain of the nucleus, and the red indicates lysine-tRNA synthetase, or antibodies specific for the synthetase. And so what you can see is that in uninfected cells, most of the synthetase, as expected, is in the cytoplasm. However, upon HIV infection, we see a large relocalization of lysine-tRNA synthetase to the nucleus. And just to quantify the extent of change, the white here is the overlap between the nuclear and the red stain. So in the uninfected cells, we only see a little bit of overlap right in the peripheral region of the nucleus, but in the infected cells, we see about a 43% overlap, supporting relocalization to the nucleus. We wanted to see whether this relocalization event occurs when HIV is heat inactivated, and also what happens with AZT treatment. And so again, here's the same experiment I just showed you. In the absence of HIV, all of the lysine-tRNA synthetase is in the cytoplasm. In the presence of HIV, we see a relocalization to the nucleus. If we use heat inactivated HIV, we do not see this relocalization to the nucleus. However, if we use AZT treatment, and that's of course a reverse transcriptase inhibitor, so it inhibits in an early stage of the viral life cycle, we see that lysine-tRNA synthetase is still relocalized to the nucleus, and this is just a DMSO control because this is what AZT was present in. So, we conclude from this experiment that it's an early event in the viral life cycle. So it happens fairly early because reverse transcription is a very early step. So I've shown you that Leu RS is phosphorylated and then goes to the nucleus upon HIV infection. When this pathway was characterized by Ehud Razin's lab, what he also showed was that treatment with a MAP kinase inhibitor, U126, inhibited phosphorylation and nuclear entry of lysine-tRNA synthetase. So we wanted to see if that would be the case, if the same pathway was being used, if that would be the case in our HIV system. And so we treated cells in the absence of HIV infection with the inhibitor to the MAP kinase, and we don't see any effect on Leu RS localization. However, in the presence of HIV-infected cells, we see a decrease in the amount of Leu RS localized to the nucleus when we treat with this MAP kinase inhibitor, suggesting that, or consistent with phosphorylation of serine 207. Okay, so we still are doing a lot of work to really understand the significance of the nuclear localization for the viral life cycle, and hopefully I'll have more on that next time we meet. But I want to turn now to the second question, and that is, once the tRNA and the synthetase are recruited into the assembling virus particle, how does the tRNA actually locate and find the primer binding site to which to anneal? And we know that the annealing occurs via the chaperone activity of the HIV nucleocapsid protein, but that doesn't explain how it gets targeted to this site. And the reason that that's an interesting question is, or question at all, is because RNA is, of course, not just single-stranded and linear like this, but it's highly structured. And so what's shown on this slide is a secondary structure of about half of the HIV genome. So each of these little dots represents one nucleotide. So the entire genome is about 10,000 nucleotides long. And what we know is that the primer binding site and the location of where tRNA is annealed is toward the 5' end of the genome. So how does the tRNA primer locate this primer binding site? And we hypothesized, based on other work in the synthetase field, that this might happen, that lysine tRNA synthetase might actually facilitate this process as well. So on this slide, I'm showing you the secondary cloverleaf structure of human lysine tRNA3. And these are the 18 nucleotides, again, that are perfectly complementary to the HIV genome. We know from early work that we did in my lab that these 2 U's are absolutely essential for human lysine tRNA synthetase binding. So it strongly recognizes the anticodon, this U-rich anticodon. So we hypothesize that maybe there's a region on the genome that, that mimics this and it allows the synthetase to interact and help to target the tRNA to the site of priming. So what's shown here is about 100 nucleotides around the primer binding site. So here are the 18 nucleotides that are complementary to 18. And located very close to that is a highly conserved stem loop, which has a loop region that mimics we felt very closely, the anticodon loop of tRNA. And so we wondered whether lysine tRNA synthetase could actually bind to this region of the genome. So what I'm showing you here is a gel shift assay where we indeed show, using this 100 nucleotide region of the genome, we show that human lysine tRNA synthetase can readily bind to this region of the genome, but other synthetases, such as human Pro RS shown here, cannot. We also wondered whether just this region of the genome would be enough for specific binding of the synthetase, and so we made what we call a mini tRNA-like element, or mini TLE, and showed that sure enough, again, lysine tRNA synthetase can bind, but tryptophan tRNA synthetase or prolyl tRNA synthetase cannot. And we also mutated these U's and showed that that abolished the binding. So we thought that maybe what's happening is that human lysine tRNA synthetase is bringing the tRNA primer into the virus particle, but then there's sort of a swap that happens and it can release the tRNA after it efficiently binds to the genome. So to test that sort of idea, we did a competition assay where we pre-bound the synthetase to the anticodon stem loop of human tRNA lysine 3 and then competed with different RNAs. And what you can see is that the mini TLE competes quite effectively, only the wild type, but not the one where those U's have been mutated to A's. Of course, the full-length tRNA can compete quite readily. But the 5' untranslated region of the genome, which contains the primer binding site, this is about 350 nucleotides, competes even more effectively than the tRNA. And we showed that the binding affinity of this region of the genome is about 5-fold higher than the binding affinity to the tRNA. And so this led to our working model that the tRNA is brought into the viral particle through, in complex with human lysine tRNA synthetase through interaction with HIV GAG. It then locates the primer binding site in part through recognition of the synthetase and this tRNA-like element in the genome. That results in binding of the synthetase to the genome and release of the tRNA so that it can be unwound and placed onto the primer binding site by the chaperone activity of the HIV GAG protein. And so this then would lead to a new hypothesis that maybe not only does the genomic RNA mimic the anticodon loop of the tRNA, but maybe there's more extensive structural mimicry. So the final thing that I want to tell you about today is our work on answering this question and really looking at the structure of the 5' untranslated region of the genome to see whether there's more extensive structural mimicry of a tRNA-like structure. So again, this is about half of the HIV genome. What we're focusing on in terms of these structural studies is the very 5' end where the tRNA binds. And on the next slide, I'm going to blow this up and show you more detail about that region of the genome. So this region of the genome has 3 really important functional elements. The so-called TAR poly A element, the TLE primer binding site region, and then shown in blue here is the packaging signal. So this is the key signal that the enzyme, the GAG protein binds to package those 2 copies of genomic RNA. So my lab has been investigating the structure of this region of the genome to answer several questions. And the one that I'll focus on today is the idea that there's a more extensive structural tRNA mimicry in this PBS TLE domain. So this, ever since HIV was discovered, structural biologists have been trying to solve structures of the genomic RNA and, of course, viral proteins by X-ray crystallography and NMR. And in the case of the RNA, the X-ray crystallography has not been successful at all, and that's in part because RNA is, again, very dynamic and conformationally heterogeneous, and it's not been possible to get crystal structures of the RNA. And so what we decided to turn to was a solution technique known as small angle X-ray scattering, which I'm sure some of you are familiar with, or SAX. And in this technique, it's important to prepare as homogeneous as possible, samples of RNA, but they don't need to be crystallized, and so that's a big advantage. The disadvantage is the structures are lower resolution. But what you get out of SAX data using computational methods are these envelopes, which are shown in gray here. So you get global structures and global conformations. We also use the SAX data in a second way, and that is we build, because you can build secondary structures and predict tertiary structures of RNA using computational methods, you can then build them and use molecular dynamics simulation and simulated annealing to then fit the models to the SAX data and refine them. And so embedded in the envelope here is shown the all-atom model that we've also built of this region of the genome. And so shown here are 2 different orientations of the primer binding site TLE region of the genome. And what you can see, as we rotate it here, is that the PBS region, these 18 nucleotides, are probably highly flexible, and they don't fit into the SAX envelope very readily. However, this region of the RNA fits quite well, and we use various tricks to show that the orientation of our RNA within the envelope is correct. And so this is the TLE, which overlaps with the anticodon region of the RNA. We also solved the SAX structure of this region of the genome bound to 18 nucleotides, which mimic the 18 nucleotides of the tRNA that anneal to it, and that's shown on the right here. So what you can see is that there's some, I don't have the overlap here, but there's some structural change in this region, but the TLE domain remains pretty constant. We also did SAX of the tRNA itself, and we wanted to see whether our hypothesis was right and whether the tRNA mimicry exists within this region of the genome. And so here I overlay the tRNA SAX envelope in black on the gray mesh of the PBS TLE domain for the apo-PBS, and then the structure with the anti-PBS annealed. You can see that there's pretty good overlap of the tRNA in this region of the genome, I think, supporting the tRNA structural mimicry that we predict. So I'm just going to summarize on this last slide here the various structures that we've solved by SAX. This is the TAR poly A element, again, the PBS TLE that I've been talking about, and then the Psi packaging signal. And so I think all of these are potential. What we're working on right now is to get larger structures of the entire domain together, and also to get structures that are with proteins interacting. And so we're very interested in getting structures of lysine tRNA synthetase bound to this region of the genome, as well as HIV GAG, which binds to the Psi region. And there are actually other proteins that have been shown to bind to as well. And so I think these protein RNA interactions, disrupting these are also potentially new antiviral strategies. So What I think, just to kind of summarize what Susan and I have told you today, is that the tRNA synthetases are actually a rich source of drug targets. So there are successful examples of anti-bacterials, for example, the one that Susan talked about against isoleucine tRNA synthetase, antifungals, this boron active compound against Leu RS, and the anti-malarial compound against proline tRNA synthetase. And what I hope I've convinced you of is that there's potentially a new generation of antiviral compounds that could be used to target these host factors, lysine tRNA synthetase and TRNALa, by targeting their HIV-specific interactions in tRNA priming and various protein and RNA interactions as well. So I just want to thank the people in my lab who did the work that I told you about today. Nathan and Alice performed all of the cell-based assays that I showed you with HIV infectivity assays. And these, Christopher, Eric, and Will have been instrumental in the SACS analysis. I've had very important collaborators over the years early on. A lot of the LeuRS work was with Larry Kleiman's work lab. And then more recently, we've been collaborating with Li Wu, who's in the Center for Retrovirus Research at OSU. Thank you very much.
So maybe I can kickstart the question. So it's really, really exciting to see that HIV virus would be able to take advantage of the lysine tRNA synthetase. So I have two questions on this. Is this a common mechanism for other RNA-based viruses also using lysine tRNA synthetase, or they use different?
So all retroviruses, actually this is how I got into the field. So I was reading literature, I was in Paul's lab studying aminoacyl-tRNA synthetase tRNA interactions, and started reading the literature on retroviruses and tRNAs. And what was, at that time, we were very interested in tRNA identity and how a specific synthetase recognizes its tRNA substrate for its normal function. And it turns out that different retroviruses use different host cell tRNAs. So all retroviruses select a tRNA, but in some cases it's tRNA tryptophan. The most common ones are tRNA trip, tRNA pro, and tRNA lyse. And there's no obvious rhyme or reason behind them. And that's one of the puzzles that we're trying to figure out. So in my lab has been studying Rous sarcoma virus, which uses tRNA trip as a primer. And we've shown in collaboration with Leslie Parent's lab at Penn State Hershey that tryptophan tRNA synthetase is also packaged into that viral particle. And we're currently working on whether these same kinds of mechanisms exist in that case. In the case of murine leukemia virus, it actually uses tRNA proline, and in that case, the tRNA proline doesn't seem to be enriched in the viral particle, and we haven't been able to find proline tRNA. So I think there's going to be some, they all use tRNAs, but whether the synthetase is involved, I think is going to be somewhat variable.
The other observation is that you have three forms of the lysyl tRNA synthetase. 60% of them are not used as a priming, but they're extremely enriched. What are they doing there?
Yeah, we don't know. So there have been some hypotheses that tRNA lysine 1 and 2 are somehow involved in the nuclear import process. And so there's been like 1 or 2 papers on that, but it's still not very clear. And so they could just be coming along from the ride and not doing anything. Probably not. You're right. They're probably in there for a reason, but I think that's still an area that...
You could also ask a question that how, translate the basic research into wonderful examples of how to connecting, curiosity-driven basic research into translational medicine.
Maybe a philosophical question. Could you imagine that if the HIV is so smart, they can find ways to be resistant against this new drug? And should it produce some special enzyme to cut it down, or?
Oh yeah, so definitely. So HIV's very clever about, so host cells often have restriction factors, and a lot of these viral accessory proteins are made by HIV to combat those and to overcome that problem. The idea behind using a host cell protein as a drug, of course, is that hopefully that couldn't mutate and and become drug resistant. So, but if there's a close interaction with, as there are in many cases, in our case we feel a close interaction between lysine tRNA synthetase and GAG. Potentially, it could still develop resistance if you're targeting that interface, which is something that we've been working on trying to do. We actually have some very successful cyclic peptides that disrupt the GAG-LysRS interaction in vitro, but they aren't effective in vivo yet. But yes, I mean, I think that's still a potential problem, but I think you have maybe a larger chance of success if you're targeting primarily, the host cell. Smart virus.
I wondered if you ever tested the antifungal, the boron antifungal, against the human enzyme, whether in fact it is also almost equally effective. You're doing a topical, so you're not so concerned about that, but I was just curious whether you've done that titration, and if so, whether you can understand it based on the somewhat differences around the active site, editing site.
So I have three answers to that. One is that Endua Wang in Shanghai, she created a yeast complementation system, and she found the human enzyme was susceptible in that situation. But we also did some work in vitro, and we found there was enough divergence that there is some species specificity for that particular boron-containing compound. Third is to remember, and Karin showed this, is that the Lysine-tRNA synthetase is part of the multi-tRNA complex. And so what happened in a yeast cell with a single Lysine-tRNA synthetase from human might be different than what's going on in the human cell. There's great selectivity, at least in the patient. And I think there's enough of a library of these boron-containing compounds that you can see enhanced selectivity for what you are trying to target versus the human. I think we could find one that interacts with the human if we wanted to, but nope. But when we did our work in vitro, what struck us as something a little bit different is that the human enzyme did not seem to rely on post-transfer editing to the extent as the Candida or the yeast Lysine-tRNA synthetase. There's another editing reaction in these tRNA synthetases that are called pre-transfer editing, and it's a very different type of mechanism. And it would use the sites in the synthetase very differently. And in Lysine-tRNA synthetase, we believe that the pre-transfer editing reaction to clear mistakes occurs in some version, some conformational change of the synthetic site. So I think also there might be an access issue that the tRNA isn't moving up to the CP1 domain. And this, I think, it is, and I think this might play off of some of the work that you showed when you talked here before, in that these different domains of more Lysine-tRNA synthetases from sophisticated organisms are adapted to do other things. So the CP1 domain might be reserved for another function.
So can I follow on that? So with so many sequence structures information available, different tRNAs, the same tRNA from different species, for example, human and pathogens. So based on the structure or even sequence, would you be able to predict whether they can be a good target to develop, highly specific inhibitors?
You can predict what happens in the laboratory, and what the chemists tell you they can make is always a challenge. At HKUST, I know we did quite a bit of that, and with some success. But then you also have to think about some other things like access and whether you can make that chemical. With Lysine-tRNA synthetase, one example that I showed up there is that the post-transfer editing site in the CP1 domain looks very different in the bacterial system and the fungal system. It's using different portions of the CP1 domain to come back and form the active site. So I think that's going to be a very powerful approach, maybe to target antibacterials. All the work that's been commercialized, or the drug that's been commercialized by Anacor actually, is a fungal drug, which is more like human.